I have experience from this space, and same type of experience is also in the board, where we also have complementary experiences for everything from business development to CMC, persons that have been involved in taking CAR T cells all the way to the market, and also regulatory expertise with a focus on biologics, which is important for a company such as Elicera. So if I take you to the next slide here to show you our pipeline of different products that we have under development, you can see that we have a diverse set of drug candidates. So 4 in total in different development stages. The one that is most advanced is ELC-100 in neuroendocrine tumors. So it's an ongoing phase 1/2 study that Magnus will talk a little bit more about shortly. We also developed a next generation oncolytic virus in 201. So this is an oncolytic virus that is applicable for treatment of basically all types of cancers. And we have genetically modified this oncolytic virus so that it elicits a 3 combined mode of action in one product. 301 and 401 are our CAR T cells under development. 301 is in liquid tumors, where we initially target B-cell lymphoma. So this is the CAR T cell that is closest to patients, so to speak. We're aiming to initiate our first phase 1/2 study by second half of next year. And then we also have a CAR T cell under development for, we've written here, glioblastoma multiforme, which is an aggressive form of brain tumor. But we also believe that the target that we're going for with 401 is overexpressed in a number of other solid tumors. Not all of them, but several solid tumors. So there's a potential outside of GBM for 401. And then, of course, also ELC-001, or the ITANK platform, which is, again, fully developed technology platform that we use to optimize our own products, but which we believe we could out-license to other CAR T developers already today, basically. So that's something that we intend to investigate the possibilities of starting this fall.
Jamal, there is a small question here, if I may.
Yeah, sure.
Yeah, it's from Per Olof. He asked here, this is stated, there's a statement on your web page, although it's too early to draw any conclusions from the study, data reported so far by Uppsala University show some promising signs of efficacy from one of the patients. Yeah. Could you explain a little how promising it is?
Sure. So important to be very clear here that this study is performed by Uppsala University, so they are the sponsors of this study. It's with IP that, of course, belongs to Elicera. It's in our product portfolio. So this is a small study where we perform this in 2 steps. And the first step is a dose escalation study. So what we're after here is to test see if we can find the maximum tolerable dose for these patients to then test in step 2 in an additional 12 patients. So we've treated 7 out of these 12 planned patients in step 1 so far. And Uppsala University has then reported that one of the patients has shown partial response, which we find is very encouraging, since this is very few patients that we treated so far, and it's also with the lower doses, so to speak. So you can hopefully expect that the chances of efficacy increases as you increase dose. So that's why we say that it's promising. But again, also important to state that it's still early days. So only treated about half of the patients that we planned so far. And we hope to be able to read out efficacy, of course, when we go into step 2 and test the maximum tolerable dose in those patients. So we'll see where we end up then.
Are you scaling this study as well, so you get an increased amount of patients with Uppsala, or?
It's already set then. So the study will be performed with the 12 plus 12 patients. Okay. And the first 12 patients are also divided in 4 different dose groups. So it's very clearly planned out, yeah.
Thanks a lot, Jamal. Carry on.
Yeah, so the slide that we're looking at here it's quite busy, but I wanted to try to catch everything as an overview with regards to what problems there are today and how Elicera addresses these problems. So I think it's easier if we just go, row by row. It makes it a little bit easier to understand, maybe. So if we start with immuno-oncology, first, it's important to state that immuno-oncology is a very broad term. So when you say immuno-oncology, it means that you use the patient's immune system to fight cancer, but this can be done in many, many different ways. I won't go into too many details here, but you can also say, as a general overview of the different ways of treating the patients with the immune system, is that you can either use therapies that helps the immune system, that inhibits the tumor's defense against the immune system. These are checkpoint inhibitors. And they work very well in a certain number of patients, but only if these patients already have a pre-activated immune system. So if there's no activation of the immune system, then these checkpoint inhibitors don't really work so well. So there are two different ways. One is to work with the brake, so to speak, and the other one is to work with the gas. And what has been shown now is that treatments in immuno-oncology have a curative potential. But to reach this curative potential, most likely there needs to be the right combination of treatments. So preferably one treatment that helps the tumor to open up to the immune system, and then another treatment that pushes the immune system to fight cancer. So monotherapies are usually not enough, even though we see that they sometimes actually are with CAR T cells in liquid tumors. But combinations, everybody more or less agrees that combinations are required. So Elicera develops CAR T cells and oncolytic viruses that can be combined with other immunotherapies. But again, we have also genetically modified these so that we can trigger a second mode of action through activation of the patient's own T cells. So you get a broader attack on cancer. If we go to the CAR T cells, so I will go through shortly what this is. Very, very short here. It's basically the T cells are the patient's immune system soldiers. So you take out T cells, you genetically modify them so that they express a CAR molecule that then can target cancer cells. This has been shown to be able to cure certain patients with liquid tumors, but there are certain challenges still remaining in solid tumors. And so a solid tumor is much more difficult to treat than a liquid tumor for different reasons. The two main ones maybe is that you have a hostile tumor microenvironment in the solid tumors, which you don't have in liquid tumors. And there's also an antigen heterogeneity that you have to take into account when treating solid tumors. So there's a lack of good targets in solid tumors, and you don't have this same issue in liquid ones. So the ITANK platforms basically meets challenges 1 and 2 here. So we, through the ITANK platform, we both get stronger T cells, CAR T cells, but we also get activation of the patient's own CD8 positive T cells against a whole repertoire of relevant tumor antigens on the tumor cells. Oncolytic viruses are viruses that selectively infiltrate and replicate only in tumor cells, but not in healthy cells. This has shown to also have effect in cancer, but the monotherapies are likely not sufficient, so you need to combine these. Again, what many are looking at now is to combine with the checkpoint inhibitors. This is something that we could do also, and we're of course interested in trying that as well. But I also want to highlight again our next generation oncolytic virus in 201, which we have genetically modified so that it expresses 3 combined mode of actions in one product. I realize now that the description I just gave in the last slide maybe makes this slide somewhat not necessary, but this just basically tells you the problems that I presented with the hostile tumor microenvironment and antigen heterogeneity that CAR T cells are faced with in treating solid tumors. And also that a CAR T cell is usually a T cell that you take out from the patient. You genetically modify it so that it expresses a chimeric antigen receptor. This then recognizes a certain target on the tumor cell. You expand these in test tube and you re-inject them into the patient. And the hope is that they will then find cancer cells and start killing them. And this type of therapy was named Advance of the Year 2018 by ASCO, one of the world's largest cancer organizations. due to the amazing efficacy that has been shown in liquid tumors, where patients that have been previously very difficult to treat, they have been able to cure them actually with CAR T cells. But again, there's still a lot to be done in the solid tumor area, and this is where we hope to make the biggest impact in the future with Elicera and our iTank platform and products under development. So this is also to try to put Elicera a little bit more clearly in the landscape of different CAR T cells under development. There are many companies developing CAR T cells throughout the world, over 100 of them. It's a rough 50-50 split between US and China, a little bit more in China. In Europe, there's only 14 companies developing CAR T cells. In Sweden, we're more or less the only ones that are doing it. So it's quite rare. We can also mention that CAR T cells have been developed in different generations. So first generation CAR T cells were shown to have an insufficient proliferation, a very brief T cell survival, and also insufficient immune activation. So for this reason, second generation were improved, then you had third generation, and then finally fourth generation CAR T cells. So they've continuously become better and better. And what a fourth generation CAR T cell is basically a T cell, the CAR T cell that contains a transgene that codes for a certain substance that can trigger the immune system. And usually they code for one cytokine that triggers the immune system. And what differs our fourth generation CAR T cells from other fourth generation is that we don't have a transgene that codes for one cytokine. We have a transgene that codes for a neutrophil activating protein, short NAP here. And the difference is that when NAP is activated, it releases a whole set of different chemokines and cytokines that together we believe give a much broader and stronger immune activation of the immune system compared to other fourth generation CAR T cells. We can also mention that still the absolute majority of CAR T cells under development are still second generation. Very few are fourth generation. And of these, we believe that most of them are actually being developed in academia. So it's quite unusual to have commercial development of fourth generation CAR T cells. There is one other company we have identified in Japan that has a similar platform technology that they've been able to successfully enter licensing and collaboration agreements with 8 different companies during the last 2 years. And this shows you the interest that the iTank platform should be able to generate. So I'll stop at that and I'll hand over to Magnus to discuss a little bit more details around how the iTank platform and our drug candidates work.
Okay, thank you, Jamal.
Sorry, Magnus. Maybe it's more a question for you, Magnus, but I know you're both very capable of answering. There's a small question here from the audience about these cytokines and the side effects. And is that something we really should be aware of? Because it looks like with your product, the fourth generation, you take a major jump compared to the competitors, as you mentioned, Jamal. But just to dive a little into the cytokine area, whether it's you or Magnus, it's up to you.
Yeah, I can maybe try to answer that. So
yeah.
You're perfectly right. Can you hear me?
Yes.
Yes, yeah. We hear you, Magnus.
So yes, you're right. So when blood cancers or liquid tumors, as Jamal are mentioning them, are treated with CAR T cells, patients can sometimes experience what is called a cytokine release syndrome or a cytokine storm that can cause side effects. Today, this is managed by corticosteroids. You can dampen the immune system and you can use an antibody against the interleukin-6 receptor. So these are manageable side effects in the clinic. It's true that when we add NAP to our CAR T cells, we can potentially induce even more cytokine, even a stronger cytokine storm. And it also means that when we go into patients with CAR T cells that have this iTank technology, we need again to start at a lower dose level to show safety. That's always the case with new products. We need to show safety first.
Good.
I hope that answers the question.
Yeah, for sure. Thanks a lot.
Yeah. Okay, so I'll take over then after Jamal here. And the first slide here is about the iTank platform. And the iTank platform is a technology to enhance immunotherapies in general, but mainly to arm immune cells in their fight against cancer and to arm oncolytic viruses, as we are doing. As illustrated here, that to the left, you see a CAR T cell with a NAP gene inside, and NAP is the same thing as the I-tank in this case then. When the CAR T cell meets the tumor cell, there will be a signal translated into the CAR T cell to start secrete this NAP transgene. And NAP has a very broad potency. It can activate the host's immune system, the cancer patient's immune system. So you can recruit NK cells to come and attack the cancer. You can recruit neutrophils to come attack cancer cells. And you can also recruit what is called dendritic cells, illustrated as a DC here. They come to the tumor in an immature form, and NAP can then mature them to become activated. And the dendritic cells, these are the teachers of the immune system. These are the school teachers. So they will travel to the lymph nodes and they will educate what is called CD8 T cells, or killer T cells, or sometimes even referred to as serial killer T cells, because they can kill one tumor cell after the other. And the idea here is that with this technology, we should not only activate the CAR T cells against the tumor. We should also induce a bystander immune response by activating endogenous, non-engineered T cells in the patient's body to activate cancer. And it's seen on this slide why we need to do that. And as it's illustrated here with the tumor, in solid tumors, antigen targets for CAR T cells are not going to be that homogeneously expressed on the tumor. So there will very likely be tumor cells escaping the therapy. And therefore, it's extremely important to induce this bystander immune response. And I think that we can do that with a NAP. So we can then activate, as you see to the right here, serial killer T cells, CD8 T cells, that can identify different targets in the tumor, illustrated by these different colored tumor cells here. So we hope to get a very strong bystander immune response, or epitope spread, as it's also sometimes called. And I think this may be the answer to how we can advance CAR T cell therapy, especially for solid tumors. The first trial that we are going into, though, is going to be with lymphoma. And lymphomas and leukemias, they are related to as liquid tumors. Especially leukemia is a blood cancer, the cancer cells in the blood. Lymphomas, then you have tumors in lymph nodes and maybe sometimes in cutaneous tissues as well. And as seen here, we can manage lymphoma patients quite well with commercial CD19 directed CAR T cells. And CD19 is a target that is expressed on all the malignant B cells, all the lymphomas in this case. And as illustrated in this product, in this slide, is that if you start with 10 patients to the left here, we are able to produce their own CAR T cells in about 9 out of 10. So in about 10%, there is a manufacturing failure, as you can see here. And when you start the remaining 9 patients, 3 will not respond. They will be primary resistant to the tumor, but you will have a response maybe in 6 out of 10. They will relapse, as illustrated later on, and some of the tumors, they will relapse and no longer respond to the CAR trial, even though they still express CD19, so they're antigen positive. But in some cases also, you will have relapses in patients, and the tumors have then downregulated the CD19 target. So CD19 CAR T cells are no longer effective. So in the end, if you go to the very right here, about 40, maybe 50% of the patients that are treated today with commercial CD19 CAR T cells. And these are very, very recent approved treatments in Europe and in Denmark and Sweden. But still, some of them will relapse. And then we think that if we are then targeting a different antigen, CD20, which is as ubiquitously expressed as CD19 on these CAR T cells, we can also treat patients that are failing conventional CAR T cell therapies. And then we hope that by arming these CAR T cells with a NAP transgene, with the I-tank technology, we can have a response also on these patients that are not responding to CD19 CAR T cells anymore. And if we can do that, that would be a real breakthrough. That could be maybe the biggest potential gain in Elicera's portfolio if we can treat those patients that, and have responses there. It can be really good. As you see at the lower part of the slide here, we are now entering soon. We are in late 2021 here. All the preclinical development is ready. We are currently producing a clinical grade retrovirus at BioNTech in Germany. And when we get this virus in the first half of 2022, we can also finalize the clinical protocol, submit an application to the Medical Product Agency in Sweden and to the Ethics Board, and hopefully get approval to start a clinical trial by the end of 2022. And as Jamal mentioned, the team here in Uppsala, we are the European team that has treated most patients with CAR T cells. We are more than 40 patients treated now so there we definitely think that we can manage to get this into a clinical trial. And hopefully we can, within a few years then, deliver good news also and therapeutic responses. We are also developing CAR T cells against a target called IL-13 receptor alpha 2, and this is especially expressed by malignant brain cancers, so-called glioblastomas, which is a really devastating disease that there is virtually no one that survives the very aggressive form of melanoma. It is going to be a little bit different challenge because in this case we will most likely inject the CAR T cells into the brain, into the cancer, rather than give a systemic treatment. The target, these CAR T cells will also be armed with a NAP transgene with ITANK technology, because here it's going to be even more important to have a bystander immune response. If you think about lymphoma, they are relatively, the CD19 and CD20 expression is relatively strong and homogeneous, but you can anyway develop resistance to these antigens. When we think about glioblastoma and other solid tumors, we will have already from the beginning an antigen heterogeneity, so we will not be able to target all the cancer cells. But hopefully with ITANK technology, we will be able to do this. So here we have selected the CAR that we want, and we are ready to start produce a retrovirus for transduction of CAR T cells. So we are 1 year behind, more or less, the ELC-301. But also here we hope to get into the clinic by late 2023 or beginning of 2024. And then we have oncolytic viruses. Jamal has introduced them. The idea here is that you will fool the immune system to believe that cancer is something foreign that need to be attacked. So you basically introduce viruses into tumor cells, because we know that the immune system is extremely potent in recognizing virus infection. So we kind of want to trick the immune system to believe that the tumor is foreign and need to be cleared. So these viruses, they have two modes of action. They can multiply inside the cancer cells, and when they are large in number, they will burst the cancer cell. So you have an oncolytic cell death, and you will release new viruses that can kill neighboring cancer cells. But this oncolytic cell death is also a strong, potent inducer of the host immune system. So you will attract immune cells like cytolytic T cells or the killer T cells to come and attack the cancer. And here we have two product candidates, ELC-100, which is not armed with the ITANK technology. It was kind of developed before. So here we are focusing very much on what is called neuroendocrine cancers, neuroendocrine tumors, NETs. And we are reliant on this two mode of action, the oncolysis, killing by the virus, and the activation of the immune system, especially T cells. And then we have the most recent development in Elicera is called ELC-201, and it's an oncolytic virus that we haven't really focused on a specific cancer indication. We believe that we can use it in many indications. So it's potentially a very broad and good virus in this sense, because it can be used for many cancers. We are in phase 1 clinical trial, as Jamal already mentioned, with ELC-100, or Advance as it's also called. And as you can see here, it's a dose escalation study. Step 1 consists of 12 patients, where we have treated 7 now, so we are at the 3rd dose level. You can see the dose level increment here. And if the patients can tolerate the highest dose level, as you can see here, which is 1 × 10^12 virus injections, we will continue in a step 2 and treat 12 more patients and look more specifically for efficacy. If the virus is toxic at this highest dose level, we will go down to the 2nd highest dose level, and we will use this dose level for the step 2. But we don't know that yet. But we are treating patients. We are including patients as we speak, and we hope to advance also this and be able to deliver some reports on this within a year and a half, I would say. ELC-201, it's brand new. It has only been, we are finalizing, I should say, the preclinical development here, and we haven't really decided yet what indication we will start with. But it has a very broad potential, I would say, and it's a very interesting candidate. So with that, I thank you, and I give back again to Jamal. I don't know if you want to say anything about the organization at Elicera, the board, and these kind of things. So I should just mention then that I have a double affiliation. I am a professor at Uppsala University and involved in immunotherapy clinical trials there, and the invention I have done together with my colleague Diju, we put all the assets into Elicera. So everything that we do here goes into Elicera, and potentially also more therapies in the future will be fed into Elicera like that.
Very interesting, Magnus. Just for the viewers, so the Oncolytic virus is your most progressing project. This area is smaller and smaller than the CAR T cell area. We know that there's at least for the first indication you have, we know there's a competitor out there or another project a little further down the road. How do you differ from this player? I think it's called Untitled Chapter, the company that look into this area. And secondly, about the CAR T cells, there's a project or a product on the street today called DARZALEX. How does that differ from the new kind of therapies that you and other guys are looking into, and especially your technique with the NAP on top of that? So that's 2 questions. I hope you can answer them in a fairly normal manner.
Yeah, so both oncolytic viruses and CAR T cells are advanced therapeutic products in a sense. Oncolytic viruses is a little bit easy because you produce a virus. It's more complicated than to produce a small molecule or a drug. But once you have it, you use it as a treatment. And I think that the company you are mentioning, is it a US-based one working also with a virus against neuroendocrine? Is that the one that you're referring to?
Yeah, that's right. That's right, Magnus.
Yeah. So that's also interesting. They have taken a different approach. They are using systemic injection. They want to treat metastatic NETs that way. Their problems right now is that since the viruses are so immunogenic, you very quickly you induce an immune response against the virus. So a second injection is useless in that case. So that is what I have reported in clinical trials. So they will most likely also have to go for intratumoral injections, as we are doing with our viruses, because we can then, we treat 4 times before we evaluate. So with the oncolytic virus field, I think that they will also change towards intratumoral injections. I think that is the way that we have to do with oncolytic viruses. When it comes to CAR T cells, as you mentioned, development has been very quick. The first CAR T cell was approved in the US in 2017. There are now 5 on the market already. 4 out of them are targeting CD19. And I should mention, which can be interesting for a company like Elicera, is that CAR T cells, they are approved after phase 2 clinical trials. There are no one's going to ask you for comparative studies with other CAR T cells. If you have an effect, you will get an approval.
Okay.
And there is one also now against multiple myeloma. It's another target called BSMA that is used. There are other companies developing CAR T cells against CD20, so we're not unique in that sense, but we are unique in the sense that we have the iTank technology in our CAR T cells. And I think that will be the factor that is making a big difference in the end. To start with CD20 or CD19 CAR T cells, it's not going to make a difference. What is going to make a difference is that you need to induce bystander immunity. And I think that we are probably the most advanced with these kind of technologies at the moment.
So if we step a little back to Agneta's presentation, that would actually drive down the health economics. So of course, it's probably in the beginning more costly to develop a more advanced product, but in the end, you might have a better economy when you look into the total treatment of the patient. Can you conclude that, or is that wrong?
Yeah, I think that is the case. And so, as you know, with all these kind of treatments, in the beginning, you have to start with the sickest of the sickest patient. You have, you cannot take away any medication that is treated, and then step by step, you are moving ahead. And what is going to be presented, I think, in a couple of weeks at ASH, this American Society for Hematology, is that CAR T cells they have an effect as second-line treatment. So with lymphoma, you get something called the R-CHOP. It's an antibody against rituximab, against CD20, combined with chemotherapy. So that will still remain first-line treatment. But CAR T cells will next year advance from third-line treatment to second-line treatment.
Wow.
And then you're right. It is initially an expensive treatment, but it's a one-shot. So if you cure them, you cure them.
Yeah. Thanks a lot, Magnus. And thanks a lot, Jamal. If you don't have anything other to add, really interesting presentation, nice and short. We have a small break. I hope you stay on, and then we give the word to XNK Therapeutics. Thanks a lot, both of you.
Thank you. Take care.